Hey guys, welcome back to my channel. This is Dr. Rajeshwar from YR Pharma Tube. In the previous video, we learnt the oxidation of benzylic carbon atoms, oxidations at allylic carbon atoms, and oxidation at the carbon atoms alpha to the carbonyl and imine groups. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of drug metabolism and medicinal chemistry, click on the links given in the description right below this video. In this lecture, we shall try to understand the oxidation reactions involving carbon heteroatoms. Particularly, we will talk about the oxidations involving carbon-nitrogen bond. At the end of this lecture, you should be able to understand the oxidation reactions of drug molecules possessing carbon heteroatoms such as nitrogen, oxygen, and sulfur. Thus, these reactions are of mainly three types. Number 1, oxidations involving carbon-nitrogen bond. Number 2, oxidations involving carbon-oxygen bond. And number 3, oxidations involving carbon-sulfur bond. In this lecture, we will learn about the first type, that is the drugs possessing carbon-nitrogen bond. Some drug molecules contain heteroatoms such as nitrogen, oxygen, and sulfur in their structure, and the process of oxidation takes place at the heteroatom. Thus, these types of oxidations are of three types. Number 1, oxidations involving carbon-nitrogen bond. Number 2, oxidations involving carbon-oxygen bond. And number 3, oxidations involving carbon-sulfur bond. Nitrogen and oxygen functionalities are commonly found in many drugs and foreign compounds. Sulfur functionalities occur only occasionally. Metabolic oxidation of carbon-nitrogen, carbon-oxygen, and carbon-sulfur principally involves two basic types of metabolic reactions. Number 1, oxidation or hydroxylation of the alpha carbon attached directly to the heteroatom, that is nitrogen, oxygen, or sulfur. See the reaction mechanism. The resulting intermediate is often unstable and decomposes with the cleavage of the carbon heteroatom bond. Oxidative nitrogen, oxygen, and sulfur dealkylation as well as the oxidative deamination reactions fall under this mechanistic pathway. And number 2, oxidation or hydroxylation of the heteroatom, that is nitrogen and sulfur only, for example N-hydroxylation, N-oxide formation, sulfoxide, and sulfone formation. There are three major oxidative biotransformations that are available for amines and amides. Number 1, oxidative deamination. Number 2, oxidative N-dealkylation. And number 3, N-oxidation. The specific biotransformation often depends on the nature of the other functional groups that are attached to the nitrogen atom. It is to note that primary, secondary, and tertiary amines are often metabolized differently. The primary route of oxidative metabolism for aromatic nitrogen atoms is N-oxidation because they cannot undergo oxidative deamination or oxidative N-dealkylation. Quaternary heterocyclic nitrogen atoms can undergo N-dealkylation but not oxidative deamination or N-oxidation. Number 1, oxidative deamination. Deamination is a chemical process in which an amine group is eliminated from a drug molecule. In oxidative deamination, amines containing alpha methyl group are metabolized by the cytochrome P450 enzyme to release ammonia and leave the corresponding ketone. See the reaction. This metabolic biotransformation mainly occurs with primary amines. However, there is some evidence that it can also occur with secondary amines. For oxidative deamination to occur, the alpha carbon, that is, the carbon atom directly adjacent to the nitrogen atom, must be attached to at least one hydrogen atom. The general mechanism of oxidative deamination is shown here. In this mechanism, the carbon atom, that is alpha to the amine, undergoes oxidation. The resulting intermediate is known as a carbinolamine because the alpha carbon atom is now attached to both a hydroxyl, that is alcohol, and an amine group. Carbinolamines are unstable intermediates and undergo a further reaction to generate either an aldehyde or ketone, depending on whether the R1 group is a hydrogen atom or a carbon atom respectively. This reaction causes the release of ammonia or a primary amine depending on whether the R2 group is a hydrogen atom or a carbon atom respectively. For example, conversion of amphetamine to its keto derivative. In this example, amphetamine is deaminated by forming the ketone. The ketone formed in this reaction is a phenyl methyl ketone, which is also known as phenyl 2-propanone. As with dealkylation, oxidative deamination involves an unstable intermediate hydroxylation step with the subsequent decomposition to yield the final products ketone and ammonia. The other examples of drugs, baclofen and atomoxetine, can also undergo oxidative deamination to form the corresponding aldehyde metabolites. In looking at these examples, note that the primary or secondary amine is removed and that the resulting metabolite retains the remaining portion of the drug molecule in the form of aldehyde. Number 2. Oxidative N-dealkylation. Dealkylation is a chemical process in which an alkyl functionality is removed from a drug molecule. This metabolic biotransformation can occur with secondary or tertiary amines or amides. As shown in the figure, the alkyl group present in the drug molecule initially undergoes oxidation to form a carbinolamine. The carbinolamine then undergoes a reaction leading to the formation of a N-dealkylated drug molecule and either an aldehyde or a ketone depending on the alkyl group that is removed. The removed alkyl group must contain a hydrogen atom. Thus, the mechanism of oxidative N-dealkylation henceforth denoted simply as N-dealkylation. N-dealkylation of tertiary amines produces secondary amines and N-dealkylation of secondary amines produces primary amines. Although oxidative deamination and N-dealkylation are similar, there is a distinct difference between these two metabolic transformations. In oxidative deamination, a carbon atom is oxidized to an aldehyde or ketone and the nitrogen atom leaves as ammonia or a primary amine. As mentioned above, the metabolite no longer contains the nitrogen atom and is composed of the remainder of the drug molecule. In N-dealkylation, an alkyl group is oxidized and removed from the drug molecule. The metabolite still contains the nitrogen atom as well as the remainder of the drug molecule. To prevent any confusion between these metabolic processes, you are highly encouraged to compare the examples which are discussed in deamination and those shown below and take advantage of the fact that deamination literally means that an amine is removed from the drug molecule and that dealkylation literally means that an alkyl group is removed from the drug molecule. In general, small alkyl groups are more likely to undergo N-dealkylation than are the larger groups. Examples of alkyl groups known to undergo this type of Metabolic biotransformation include methyl groups, ethyl groups, propyl groups, isopropyl groups, butyl groups, and benzyl groups. It is to note that due to the lack of a hydrogen atom, functional groups such as a tertiary butyl group cannot be removed by N-dealkylation. Examples of two drugs that are known to undergo N-dealkylation are shown in the figures. The functional groups on meperidine, a tertiary amine, and diazepam, an amide that can be dealkylated, have been highlighted. In both the reactions, the methyl group present on the tertiary amine nitrogen are eliminated to form their corresponding dealkylated products. There are 4 points to be considered regarding the drug molecules and N-dealkylation process. Number 1. Whenever a tertiary amine contains two different alkyl groups, as seen with the structure of nicardipine, the smaller alkyl group is almost always removed before the larger group. As shown in its structure, the larger alkyl group contains a benzyl carbon atom that is attached to a hetero atom. As previously discussed, this benzylic carbon atom can undergo oxidation, however, it does not produce a stable secondary hydroxyl group. Oxidation of this benzylic carbon atom forms a carbinolamine that collapses and forms an N-dealkylated metabolite. Thus, oxidation at this carbon atom is correctly described as N-dealkylation and not benzylic oxidation. Number 2. As exemplified by atenolol structure, drug molecules that contain secondary amines can directly undergo oxidative deamination or initially undergo N-dealkylation. In these situations, oxidative deamination is normally a minor metabolic pathway and N-dealkylation is normally the major metabolic pathway. Number 3. Secondary and tertiary amines located in the middle of a drug molecule and attached to large alkyl chains are less likely to undergo N-dealkylation. This is illustrated with the salmeterol and as an example of a previously noted concept. Functional groups that are sterically hindered or otherwise difficult to access are much less likely to be metabolized than those that are easily accessible. In terms of salmeterol, aromatic oxidation and benzylic oxidation are less sterically hindered than is N-dealkylation of the large alkyl chain. And number 4, as illustrated in the figure, amitriptyline can undergo two N-dealkylations and an oxidative deamination. Similar to atenolol, as previously discussed, once the first methyl group is dealkylated, the resulting secondary amine could directly undergo oxidative deamination. Returning to a previously discussed concept, the human body uses only the minimum number of these biotransformations that it needs to enhance the water solubility of the drug and allow for its excretion. For some drug molecules, all of these steps are required. For others, perhaps only one or two methods. Secondary and tertiary alicyclic amines can form lactones, as illustrated with niacin, which is also known as nicotinic acid. Oxidation of the alicyclic carbon atom adjacent to the tertiary nitrogen produces a carbinolamine. The carbinolamine subsequently breaks down to form a secondary amine and an aldehyde similar to what was seen with oxidative deamination and N-dealkylation. However, because this metabolic biotransformation began with an alicyclic ring, both functional groups remain as part of the initial metabolite. This aldehyde can go further oxidized to carboxylic acids by condensation and lactone formation reaction. Because the secondary amine is close to this newly formed carboxylic acid, the functional groups can combine using a condensation reaction to form a lactone. The methyl group can be removed via N-dealkylation either prior to or after lactone formation. Lactone formation does not always occur with alicyclic amines. However, it should be considered as a possible metabolic pathway. And number 3, N-oxidation. In chemistry, an amine oxide is also known as amine N-oxide or simply N-oxide. It is a chemical compound that has the chemical formula R3N plus O minus. It contains a nitrogen-oxygen coordinate covalent bond with 3 additional hydrogens and or substituent groups attached to nitrogen. Sometimes it is written as R3N2O or alternatively as N3N double bond O. In the strict sense, the term amine oxide applies only to oxides of tertiary amines. Sometimes it is also used for the analogous derivatives of primary and secondary amines. Examples of amine oxides include pyridine N-oxide, a water-soluble crystalline solid, and N-methyl morpholine N-oxide, which is an oxidant. N-oxides are common metabolites of psychoactive drugs, for example nicotine, zolmitriptan, and morphine. Unlike oxidative deamination and N-dealkylation, N-oxidation involves a direct oxidation of the nitrogen atom as opposite to an adjacent carbon atom. Although it is not necessary for an adjacent carbon atom to be attached to a hydrogen atom, the products of N-oxidation vary based on the presence or absence of a hydrogen atom. N-oxidation of secondary and tertiary amines can be catalyzed by either cytochrome P450 enzyme or flavin monooxygenase enzyme, whereas N-oxidation of primary amines is almost always catalyzed by cytochrome P450 enzymes. Primary amines that are attached to a carbon atom that lacks a hydrogen atom can be sequentially oxidized to hydroxylamines and nitroso groups. Overall, only a few drugs have this structural feature. N-oxidation of primary amines. For example, amantadine, an antiviral agent, undergoes N-oxidation, initially forms hydroxylamine derivative and then to a nitroso derivative. It is to note that oxidative deamination is not possible for amantadine. The carbon atom adjacent to most primary amines is normally attached to at least one hydrogen atom. Although oxidative deamination is the most likely metabolic biotransformation for a primary amine, it can also be directly oxidized to sequentially form hydroxylamines, imines, oximes, and aldehydes. In the other example, primaquine, an antimalarial agent, undergoes N-oxidation, forms its hydroxylamine derivative. This is then loses water to form an imine. Note that the aldehyde metabolite resulting from hydrolysis of the imine is the exact same metabolite that would be formed if primaquine were directly metabolized by oxidative deamination. The oxime metabolite is a tautomeric form of the nitroso group seen with amantadine. N-oxidation of secondary amines. N-oxidation of secondary amines can sequentially form hydroxylamines, imines, nitrones, and aldehydes. On the rare occasion that both carbon atoms attached to the secondary amine lack a hydrogen atom, then only a hydroxylamine can be formed. An example of what could occur with the N-oxidation of a secondary amine is shown with encanide. Similar to primaquine, the original hydroxylamine can undergo a dehydration reaction to yield an imine. Hydrolysis of the imine opens the ring and produces the same metabolic product that can be directly formed via N-dealkylation. Further oxidation of this imine produces a nitrone. N-oxidation of tertiary amines and heterocyclic amines. Tertiary amines as well as heterocyclic amines can be directly oxidized to form N-oxides. Shown in the figure are the structures of prochlorperazine, a drug with multiple tertiary amines, and zaleplon, a drug with multiple heterocyclic amines. A single nitrogen atom has been chosen to illustrate N-oxidation for both of these drugs. Note that this metabolic biotransformation could also occur at the other nitrogen atoms on these drugs. As with other examples of N-oxidation, there are other metabolic biotransformations that could occur, some of which may be more prevalent. In the case of prochlorperazine, N-dealkylation and aromatic hydroxylation followed by phase 2 conjugation are much more prevalent than N-oxidation. The examples here are provided to illustrate metabolic pathways that could possibly occur. In some instances, N-oxidation can lead to the formation of potentially toxic metabolites. This is especially true if adjacent functional groups help to catalyze the formation of a highly electrophilic intermediate. A good example of this is seen with acetaminophen. When taken acetaminophen in normal doses, approximately 1 to 2% of the drug undergoes N-oxidation. Due to the presence of the paraphenol group, there is a subsequent dehydration and the formation of a highly reactive electrophile. At normal doses, the concentration of this reactive intermediate is low enough that it is readily deactivated by glutathione conjugation. However, in acetaminophen overdoses, saturation of conjugation pathways can lead to liver damage and death. This is the list of references followed for the lecture. That's all in this video, the oxidations involving carbon-nitrogen bond. In the next lecture, we will discuss about the oxidations involving carbon-oxygen bond and carbon-sulfur bond. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.